So before we can start getting to the stats, we have to introduce some common terms and introduce also how to work with our data, the very first steps of working with our data. So what we're going to do is start with some of the definitions with data in biology. So there's really two main fields in statistics. There's descriptive statistics and there's inferential statistics. Descriptive statistics is the organization and summarizing of the data. In descriptive statistics, what you're trying to do is take your data set and either display it visually as a graph, a figure, or a table, or summarize it using numbers, such as means and medians, ranges and variances, etc. With the descriptive statistics, you're not trying to do any sort of statistical test. You are just trying to describe the data. What's the central tendency? What's the spread? With inferential statistics, you are trying to draw conclusions from the data. With these conclusions, you generate p-values that kind of give you a measure of confidence. If you have a high degree of confidence in your analysis, then the conclusions are probably correct, but they could be wrong. If you have a low degree of confidence, then your conclusions are probably wrong, but again, they could be correct. I mention this, they could be correct or they could be wrong, because as we get to the statistical tests and talk about error, we have type 1 errors and we also have type 2 errors, and those errors kind of quantify this aspect of they could be wrong, they could be correct or they could be wrong. When we do inferential statistics, we rely on probability theory. Probability is the likelihood of an event occurring. A lot of this was developed in the past. So for example, Carl Pearson was pretty big in the field of statistics and biology, and we mentioned him. When he developed some of his theories, such as binomial theorem, he flipped a coin 24,000 times just to record the outcomes to see if his predictions actually matched what the math tells him. Out of that 24,000 times, he obtained 12,012 heads. Pretty darn close to 50-50. So when we work with statistics, we've got two different groups. We've got the sample and we have the population. We're going to start with the population, which is basically the universe. Our textbook definition is that the population, textbook definition of the population is the totality of individual observations about which inferences are to be made, existing anywhere in the world, or at least within a definitely specified sampling area, limited in both space and time. This is basically the group that we are trying to make conclusions or draw conclusions from. This population doesn't have to be the entire world. We can make it arbitrary. So for example, if we wanted to make conclusions about roaches in the Cavness building, then we would define our roaches as those being in Cavness building. If we wanted to know about roaches on ASU's campus, then we define that as ASU's campus. If you wanted to study roaches in the state of Texas, we can define it. Now, why is this important? Well, once we define our population, that then leads us to the sampling protocol, because what we want to do is try to generate a sample that is representative of our population of interest. We wouldn't collect roaches only from Cavness if we were interested in describing the roaches throughout Texas. I will note that our population could include subsets of experiments. So if we do an experimental study and we manipulate some single variable and we have 5 replicates, well, our population in that case is all possible replicates that we could have done if we had unlimited time and resources. Also note that most populations are finite, so there's clearly defined boundaries, but when working with probability, we assume that those populations are infinite. For the population, when we describe some of our statistical terms, such as means and medians and variances, those descriptors are called parameters. So population parameters. It's these parameters that we try to estimate when we generate a sample. The reason we generate the sample is because it's too unlikely that we would collect every single individual in our defined population. So, in order to make estimates or inferences about that population, what we would do is try to draw a random sample. If we draw a random sample, then frequencies within our sample should equal the true frequencies in our population. In this case, our sample would be completely reflective of the total population that we are trying to make inferences about. With our samples, we can calculate the same types of statistics, so means and medians and variances and etc. But now, instead of being called parameters, we refer to them as statistics. So I list sample statistics here. We could also just say regular statistics. So with samples we have statistics, with populations we have parameters. How we generate our random sample is going to be based on the population. If we know the entire population, then perhaps we could go out and sample every single individual if we have unlimited time and resources, but usually we don't. So one way to collect a random sample is to assign each individual a number, then randomly pick the numbers, and you then go out and measure those individuals. Most likely scenario is that we don't know the entire population. So what we could do is use the computer to select a random number of points, and then at those points we establish grids and we take all of our measurements from those grids. Another way of collecting random samples is to pick a random point and then randomly choose a direction and start walking a transect. Now these examples for doing the studies, it's all about if we were out in the field and measuring, let's say, plants or trees or whatever in a forest. If we were trying to do random samples of animals, then we can also do kind of the same thing. We could set up random places and collect our animals at those places. Inside our random sample, they consist of observations. Now there's going to be two types of observations, or at least observations fall under two types. We have the individual observation and we have the sample of observations. Well, the sample of observations is just that. It's the sample. It's going to be our data set. This is the data that we're going to summarize, that we're going to use to make inferences about our population. So for our roaches, if we went out and measured the wing lengths of 50 roaches, then all 50 wing lengths represent our sample of observations. We also have individual observations. That is the data point. That is that single item. So out of the 50 roaches, each roach produced an individual observation. So each roach produced a single wing length. When I refer to sample, I'm generally talking about the sample of observation. So, and fortunately others do as well. So trying to distinguish between an individual observation and sample of observations, it's not too difficult. Now each observation is going to be either a measurement on that individual, or it's going to be some sort of attribute. So the measurement is wing length, attribute could be color, or it could be, let's say, size, just small, medium, large. Our measurement variables, or our attribute variables, is the property that we measure on our individual observations. All right. So our variable could be measurement, or it could be some attribute. When we work with a variable, our rule of thumb when creating data sets is that each column in our data set represents a single variable. All right. So for the roaches, if we measured wing length and let's say tarsus length, wing length, that's a variable that have its own column in R and Excel. Tarsus length, that's another variable. That's going to have its own variable in R and in Excel. So each individual has a single measurement from it for each variable, so wing length, tarsus length, but each individual could be measured multiple times. So we measured both wing length and tarsus from the same individual. Now that roach now becomes the smallest sampling unit. Now, why do we worry about what the smallest sampling unit is? Well, it's because when we analyze our data, we assume that our data are random and independent of each other. One of the ways in which we ensure that is if we have one measurement variable per sampling unit. So, if all we did was measured wing length on the roaches and analyze wing length, then there's no problem. Each wing length is independent of all other wing lengths because we've measured different individuals. But if we try to analyze both wing length and tarsus length, now we have more than two variables on that same sampling unit, and our variables are not independent. You can say why. You can ask why is that? Well, think about it. If we have a large roach, it's likely that they also have larger wings and larger tarsus lengths. Small roaches have smaller wing lengths and smaller tarsus lengths. So the wing length and tarsus length are probably correlated to the size of that individual. If they're correlated, then by definition, they are not independent of each other. So if we do have that situation, that actually now represents a bivariate sample. We have paired observations on each sampling unit. Can we still analyze them? Yeah, we can. Can we do our typical statistics, so T-Tests, Anovas? No, we have to account for that non-independence between our two different variables. Another example is heart rate. So if we measured heart rate before exercise and heart rate after exercise, well, heart rate before exercise is a variable, heart rate after exercise is another variable, but we've measured them on the same individual. The individual is our smallest sampling unit, so in order to analyze the effect of exercise on heart rate, we have to account for that non-independence. So just as a review for the samples and populations, our population is the universe. This is all of the individuals that we want to make inferences about. By convention, when we refer to individual data points in our population, we use capital letters. So we have item 1, item 2, item 3, item 4, and then all the way up to however many individuals or data points we have in our population. We assume that the population is infinitely large, or at least large enough to where we can't measure everyone. So what we do is have a sampling method to generate a random sample. With our random sample, each data point is now represented by a lowercase. So here's item number 1, item number 2, number 3, number 4, all the way up to our total sample size. Note, the n's differ in capitalization as well. This is just to emphasize that this number, whatever, maybe this is item number 1,000, this number isn't necessarily item number 1,000. This could be item number 50. So you can have different sizes of data points in our population versus the sample. I mean, after all, that's why we generate a sample. From our sample, then, we try to infer information about our population. How do we make inferences about them? Well, usually it's about the sample statistics. So with our sample statistics, we have a mean, a standard deviation, a correlation, a difference, and a proportion. When we refer to the sample statistics, we utilize regular letters, English letters. I believe they could be called. Not sure what other term we could use. We'll just say English letters. That represents standard sample statistics, and what we try to do is infer the population parameters. When working with population parameters, we now utilize the Greek letters, so mu and sigma and rho and delta and pi. Ideally, if we have a good random sample, then our sample statistics should approximate the population parameter, and we'll talk about that in a bit. Alright, so now that we kind of understand samples and populations, we can move on to the individual variables. Why worry about variables to begin with? Well, the variable number and the variable type determine what type of statistics we can perform. When doing science, our goal is always to select a variable whose property varies or differs in some measurable way. We need to be able to detect and measure that difference if we're going to have a successful research project. When thinking about these variables, we can classify the variables by type and by scale of measure. So we'll start with variable type. This is just basically the classification of variable. We can have a qualitative variable or we can have a quantitative variable. Qualitative variables are non-numeric data expressed by using their attributes. These are also called categorical variables. They're also called nominal variables. You will hear me use both categorical and nominal variables throughout the course. Examples would be hair color, or sex, or nationality. These variables are just the attributes. They're the categories. When we work with these data, we often present them in combination with frequencies. So we would generate a frequency table or perhaps even a bar graph just to say this is the number that we counted in our sample. Perhaps this is the percentage of each category in our sample, and so forth. Now, while these are attributes, sometimes we convert the attributes to a number just to make it easier to work with in our data set. So using if functions in Excel or selection functions, sometimes it's easier to work with numbers instead of the actual term. So even though they are numbers, each number represented a category, and that keeps them as a qualitative variable. The other variable type is a quantitative variable. Quantitative variables are those that are expressed numerically. Now, this isn't the conversion where we use numbers to represent a category. These are actually measurement variables. We went out and measured them in some way, either measured them or counted them, and their measurement or count is a number. So of these quantitative variables, we can have continuous or discrete variables. Continuous variables can take any number between our two fixed points. So we have an infinite number of possibilities. If we measured height, well, height is a measurement variable. It's a continuous variable. We can have any number of points between, let's say, 0 and however large our organism can grow. Now, we would say things like, well, it's 30 inches or it's 40 inches. Yeah, that's we've rounded. But if we were going to be really precise, we can go out to 1 million or more decimal places with our height value. So every individual probably has a different height value. When we round, though, then we start getting repeats in our data set. So what you should think about is when you measure, could I increase precision with different instruments? If yes, then it's likely a continuous variable. Discrete variables differ in that you can't have an infinite number of values. Your actual values are going to be restricted to specific numbers, usually whole integers. All right. So when we work with discrete variables, they can also be called discontinuous or meristic variables. I probably won't use either of those terms. I will probably use discrete throughout the course. So a discrete variable would be a count. You know, how many leaves are on a tree? Well, you count the leaves, and you don't normally measure a half a leaf. They either have a leaf or they don't. Clutch size, when you count offspring, you can't have half a child or half an offspring. You can't have 3/4 of an offspring. So those are all discrete variables. We are confined to whole numbers. The other aspect of our variables is the scale of measurement. This is kind of important because if you do a test that makes an assumption about the scale and you're working with the actual wrong scale, then you could make mistakes in your interpretation. In reality, is it a big deal? Not really. There's a few cases where it matters, but overall, what's most important is the type of variable. That determines our test, and then we can use scale just to confirm that our assumptions are correct. So what are the 4 types? Well, we're going to start with the first 2 types, nominal and ordinal. These 2 types only pertain to qualitative data, so those that are expressed by categories. The difference between nominal and ordinal data is in the ordering. So if our attributes have a natural order or an implied order, then we're working with an ordinal variable. If they do not, if they don't have any sort of order, natural or implied, then it's a nominal variable. So hair color, eye color, zip codes, those are all nominal variables. There is no order. There's no implied order. We don't say that brown hair is better than blonde hair, or blue eyes is better than brown eyes. Those are just attributes. Ordinal data actually have a natural or implied ordering. Shirt size, so if we measured shirt size as small, medium, and large, well, that has implied or natural ordering. Certain sizes, they get larger as we go up in size. Preferences, so if I asked you to rate your favorite classes, well, and you give them a number. 1, you like them. 0, you don't like them. 10, you like them really, really much, a whole lot. Then that preference is in order. 10 is good. 0 is bad. One thing to note is that with the ordinal data, there's no assumptions about the differences in the state. So if you listed your preferences, the difference between a 2 and a 3, while we know 3 is larger than 2, we don't know how big a difference there is between those two values. And the difference between a 2 and 3 may actually be larger than the difference between a 7 and 8. An example that I've encountered is working with pathology and indices. So you infect a chicken with the virus, and then during your observations you judge its health on a 4-point scale. 0, no infection. 1, you have minor discomfort. They look uncomfortable. 2, they're running a fever, sluggish. 3, severe infection. They do not respond. They don't move when you reach in to grab them. And 4 is death. So you can look at the difference between a 0 and 1 and between a 2 and a 3, and they don't have the same magnitude of difference. So with ordinal data, even though there's natural or implied ordering, there is no assumption about the magnitude of difference between each of the scales. They could vary. Interval and ratio scales are quantitative variables. Intervals, the difference between interval and ratio comes down to a meaningful 0 point. This doesn't mean is there a 0 in its scale. It means is there a real 0 that we can't go below. Alright, so interval scales are going to range from negative infinity to positive infinity. We could have some limits, but the idea is that the 0 isn't like an ending point. We can go below that. With the ratio scale, we do have a 0. We can't go below that 0 at all. Now, can we get down to the 0? Probably not, but at least we have a real 0. So height has a real 0. So if we measured height of a baby, we know it's not going to be 0. Babies are going to be born and they're going to be 10 inches, 12 inches, maybe 18 or 19 inches, who knows. But our bottom scale is above 0, but we do have a 0. We can't have a negative height at all. So that's a very defined, that is a meaningful 0 point. IQ, that does not have a meaningful 0 scale. IQ is more arbitrary. A lot of our standardized tests, those are arbitrary numbers. There is no clearly defined 0 point there. Now, what does that mean for us? Well, it comes down to this idea of ratios. All right, this idea of ratios. So for the interval and ratio scales, the differences between numbers are constant. So going from 1 to 2 or 5 to 6, that's all 1 unit. If we go from 5 to 10 and 25 to 30, we've increased by 5 units. The difference between the numbers remains the same. So that's constant. Difference is the ratio. So if we take 2 divided by 1, well, that's double. If we do 20 over 10 as a ratio, that's double. that all makes sense, and for a ratio, all of the numbers will make sense. But with an interval, if we did 20 over 10, that's 2. What do we do about 5 over negative 5? That ratio doesn't make sense. Another example of interval scale is temperature. So temperature in degrees Fahrenheit. Yeah, we can go below 0. And if we say we go from negative 5 up to 10 degrees, negative 5 low and 10 degrees for the high, well, we can say it's 15-degree difference. But if we go from 5 to 10 degrees, we would say, yeah, it became twice as warm. But what if we do from negative 5 to 0 degrees? We really can't say it's twice as warm. So the ratio doesn't make sense. Is it really a big deal between interval and ratio? Not really. Not really. Alright, so for practice, I want you to name the variable type and the scale for these following variables. I'm going to let you pause it so you can jot down your answers. What I want you to do, and this will be on quiz and on tests, I want you to give the variable type. So your options for the variable type will be qualitative, contiguous, or discrete. Those are the 3 variable types that we'll use. So qualitative, continuous, and discrete. Then for the scale, you will list nominal, ordinal, interval, or ratio. Alright? And remember your rules. Nominal and ordinal can only go with qualitative data. Interval and ratio can only go with continuous or discrete data. So go ahead, pause this video, and jot down some answers. Alright, so hopefully you've taken some time to answer this. And let's go over this now. So shirt size, I assume this is small, medium, and large. It's a qualitative variable. It's ordinal. It has a very clearly defined order. Zip code, it's qualitative. It's nominal. So if you think about zip codes, also area codes, those are just arbitrary. We use the numbers to represent different areas within the U.S., but there's really no clearly defined ordering system. We can't say one number's higher or one number's larger than the other. They're just arbitrary. They're just categories. Distance between cities is a continuous variable. We can go to infinite number of decimal places if we want. It's also ratio. We can't have negatives. We can't have a negative distance. Standardized test scores, these are discrete. Usually they're discrete. You can't have decimal points. These are also interval. For the scale, you generally can't get negative scores, and in some cases the scores are based on who else took the test with you. Leaves on a bush, I consider this discrete. You don't normally count half leaves or quarter of a leaf. It's also ratio, so you can't have negative leaves. We have a clearly defined 0 point. Grade depends on if we are working with a letter grade or a percentage grade. So if we have a letter grade, then this is a qualitative ordinal variable. The letter grades have a natural and implied ordering. If we're working with the percentage, well, now it's continuous. It's a continuous type of variable. You have decimal places, and it's a ratio. So your statistical tests that are available to you depend on if you're working with a letter grade or percentage grade. Time to complete a task, this is continuous. It's ratio. You can't complete a task in negative time. It's just not possible. And our last one is ratio of measurement variables, such as surface area to volume ratios. These ratios are continuous. You can have decimal places, but they're also interval scale. So you really can't have a 0. If you have a 0 volume, you can't. It's undefined. You can't go negative. So just as a summary, I have this table here, and it just kind of tells you what you can and can't do with the different types of variables, and especially the scale of variables. So nominal and ordinal, those are the categorical variables, the qualitative variables. When working with those, we can present counts, we can do frequency distribution, so frequency tables, bar graphs, and so forth. With ordinal data, you can calculate a median. This should be checked for nominal, or let's say half a check, because you can also do the mode, which is just the most common value. And we know the order of the ordinal variables. You can do the same things with the measurement variables, so the quantitative variables, but the quantitative variables also allow us to do several other things, such as quantifying the difference between each of the values, adding and subtracting the values, and so forth. But only ratio variables allow us to do multiplication and division, and that's because, again, these have a clearly defined zero. So when we and this isn't just multiplication and division for certain parts of our range, of the range of values that we could take, but it's for the entire range of values. So yeah, temperature, degrees Fahrenheit, we can multiply and divide when we have positive values, so when it's warm. But if we have warm values and we have super cold values, multiplication and division doesn't make sense. So it only applies for ratio, and then also ratio variables have a true zero. So key point on these variables, and this is extremely important. You need to know them. You need to know the type of variable that we have and the scale of variables. Why? It's because the statistical tests mostly depend on the type of variable that we have and the number of variables that we're working with. All right. So if we have qualitative variable, we're going to do a comparison of frequencies. That's what we're doing. We can't do a comparison of frequencies when working with quantitative data. Two different statistical tests, two different requirements. Likewise, with our quantitative variables, we can do comparisons of central tendency and dispersion. So we can compare means, we can compare medians, we can compare variances. We can't do that with qualitative data. And then what specific tests, so T-tests, Anova's, Cochran-Mantell tests, etc., all kind of comes down to how many of the variables do we have. So once we decided on what tests we could run, then the number gets us to that next step. Scale of measure has a very limited influence on the statistical tests. So with our qualitative variables, so categorical and ordinal, we're going to use the same test for both. Chi-square tests, G-tests, we use those for both regardless of the actual scale. But if you do have an ordinal variable, there are some extra tests that are available. And those tests do assess magnitude and direction. So are certain values higher than other values? Do certain restaurants have a higher score than other restaurants, and so forth? With the quantitative variables, we're going to use the same test for both types. So Anova T-Test, they can be used whether we're working with continuous or discrete variables. The biggest difference is usually in the display of how we display the data. So if we have discrete data, it doesn't really make sense to do a lot of box and whiskers plots, so doing a mean and then variances, because normally we're doing counts. That's what we have for the discrete variables. And we could talk about it. If you have questions, feel free to come and talk to me. Two other terms that we should address before we start getting into the descriptive statistics is accuracy and precision. All right, so our goal with collecting data and analyzing data is consistency. We want to keep sampling error the same for all of our analyses throughout the entire experiment. All right, so what is sampling error? That's the chance difference between the estimate and the population parameter being examined. Sampling error is all about just dumb luck. Maybe you woke up in the morning and your eyes aren't as good at spotting, the birds that you're trying to count. So sampling error is just the randomness that's out there. We also want to eliminate bias. So bias is a systematic discrepancy between estimates and the true parameter. Bias can be introduced by the the researcher itself, or it could be introduced by equipment. So temperature thermometers that you can buy in a grocery store are notorious for being biased. You take them, you should always check them because maybe they read low. So a pot of water could be boiling, you measure that temperature, and it's only reading 205 degrees Fahrenheit. Well, we know that's not correct. If you measure, water that is starting to freeze, thermometer could be reading, 28 degrees Fahrenheit, right? It should be ice at that point. That's bias. That thermometer is consistently reading low all the time. For the researcher, you can have bias if you were going out to collect and you only collected in specific areas. You're not having a random sample. You are biasing your numbers to data points that came from those specific areas. So we always try to eliminate both sampling error and bias. Now, what effect do both of these have? Well, they're going to affect accuracy and precision. So accuracy is affected by bias. Accuracy is the closeness of the measured values to its actual true value. So how close is our sampling mean to the true population mean? That's accuracy. Non-random sampling, instrumentation error could produce this bias and reduce our accuracy. Precision is how close each of our measures are to each other, so the repeatability of our measurements. Precision is affected by sampling error. If we have a small amount of sampling error, then all of our points are going to group together. If we have a lot of sampling error, then all of our points are going to be spread out. Measurement error and sample size can affect precision. So with a measurement error, it could be based on the instrument itself. So if you have a scale, a balance, especially a digital balance, you can look at the label and it'll say what the precision is, maybe plus or minus 0.01 grams. Same thing with a pH meter. It'll be plus or minus 0.1 or plus or minus 0.01. All right, that's precision. Precision could be thought of as the variance or the standard deviation. So the higher the precision, the smaller the spread of data points, the smaller the variance and standard deviation. Using targets is a good way to kind of compare these two terms. So if we are accurate and precise, then all of our points tend to hit the bullseye and they're all grouped closely together. If we're precise but not accurate, all of our points still group together, but now we miss the mark. Our samples no longer hit or no longer match the true value, our target. If we're not accurate and not precise, then yeah, we're spread out. The precision is, look at all of our points. We're spread out on the target, and if we take like the mean of these points, we're centered like right here. We miss the mark, so we're not accurate or precise. We can be accurate and not precise, be it in this graph where we have a wide spread, but if we take the average, yeah, we're hitting the target. The average is hitting the target, even though each of our individual points does not. So accuracy and precision kind of relate the sampling error and the bias in our data set, and they're directly related to our individual sample statistics. All right, so now moving on. We've covered the definitions. Now what we want to do is talk about some rounding. So we're going to do some rounding, and then we'll talk about some basic displays of our data. So why do we round? Well, we round continuous data so that we're not working with thousands of decimal places. So rounding makes things easier, and in a lot of cases when we round, we're rounding because the extra precision just doesn't really make any difference. All right? It doesn't matter if a wing length is 0.00001 millimeter different than another wing length. We can round and make our life a whole lot easier. When we round a value, our value now takes on an implied limit, which is a range of measurements that true value could actually fall. All right, so with our rounded values, when we have a rounded value and we try to generate an implied limit, we always go to one more digit. So for example, 5.64. We will assume that we rounded here at that 4. That 5.64 could represent anything, any number from 5.635 to 5.644. Any number between these numbers here will round to 5.64. Why is this important? Well, it's important when we start working with derived variables or doing ratios and so forth. The implied limits for a single variable get carried over to those other variables, and we'll see some of that. So it's important to know that when you round, each of our rounded values has an implied limit. Now, how should we round? At what digit should we round? Well, that comes down to significant figures, and for significant figures we're not going to be as stringent as chemistry is, because again, chemistry kind of relies a lot on this implied limits idea, and really with the limits the precision of the instrumentation that they use. For us, it all comes down to how many steps we have from our largest value to the smallest value. What we want to have is 30 to 300 steps between our smallest value and our largest value. This tends to give us enough precision to where we can adequately describe and detect variation in our data, but it avoids having too much precision so that instrument error starts to have a compounding effect on our analysis. I'll say that experience and practice makes this decision second nature. And when you talk to researchers, and I'm sure if you start doing a lot of research and collecting data and analyzing data, that you just kind of get a feel for where you need to round. And that feel ends up following this rule where we have 30 to 300 steps between the lowest and highest value. So how do we round? Well, we're going to follow these rules, and I'm sure you've used them before if you've rounded any type of data. So we have a target digit that we want to round to. We then go to the next digit and determine, is it less than 5, is it greater than 5, or is it equal to 5? If it's less than 5, then we leave our target digit alone. If it's greater than 5, then we round our target digit up. If that next digit is 5, then we have to do this. We have to look to see what's after that 5. If it's missing or if it's 0, we're going to leave our digit alone. We will assume that there's nothing after it. If that digit after the 5 is non-zero, then we assume that we're not quite 5, we're actually a little bit above 5, so we're going to round up. These are the rules that we're going to follow because this is what R does. But you probably quickly see that if we always do this, then we're going to probably be leaving way too many digits alone when we have 5 and nothing after it, or 5 and a 0 after it. So some textbooks have additional rules that kind of look at the actual target digit itself. So if the target digit's odd, then you round it up. If it's even, you leave it alone. And that way you have the same number of digits going up as staying the same, and so forth. We won't do that. We're just going to do this. Look at that next digit. If it's a 5, look to see what comes after it. If there's anything after it, anything non-zero at all, even if it's 0, 1, all right, we decide that it's slightly larger than 5, we're going to round up. If nothing's after it, or if it's just zeros after it, then we will say, well, it's just a 5, we'll leave our digit alone. Alright, so here's some rounding practice. I want you to pause this video, and what I want you to do is to do the rounded number to the digit that I want you to do. So round to the ones digit, we're going there. Alright, so do the rounded number and then give the implied range of that rounded number. Don't worry about the rule. I'm not going to ask you what rule did you use to round. Just go ahead and practice rounding to the digit that I specifies. Alright, so go ahead, pause the video, work on that. Alright, so hopefully you took the time to round these numbers by hand and come up with their implied range. So let's take a look at them. Alright, so the first number, we want to round here. That's our target. We look at the next number of 3. That means we leave that alone. So our rounded number is 13. The implied range always goes to the next digit. So for our implied range, we're going to keep things easy and say our implied range is 12.5 up to 13.4. So this number basically says any number within these numbers, so 12.5 to 13.4, we're going to round to 13. Next one, we're rounding there. That's our target digit. So target digit's a 5. We look at the next one, that's a 5. Well, because it's a 5, we go to the one more, it's a 1. So this is basically greater than 5. Just a little bit more than 5. We're going to round up to 0.96. Our implied range is one more digit, so it's 0.955 up to 0.964. Again, these numbers will round to our rounded number. Next one, we're rounding here. Target digit's a 1. Next digit after that's a 3. It's less than 5, so we leave our value alone. We get 0.011. The implied range is one more digit, so 0.0105 up to 0.0114. These numbers round to this one. Next one, that's our target. We look at the next number, it's a 5. There's something after the 5, so our 5 is actually a little bit more than 5. We round up. Alright, so this goes up to 0. We bump the 1 up to 2, so our rounded number's 0.20. Now note, I include the 0. That's important because it emphasizes that we rounded at this point. We didn't round at the tens, we rounded there. That has its implications for the implied ranges. So the implied range for this is 0.195 to 0.204. Again, these numbers round to this. If we left off the 0 and said it's just 0.2, well then it changes our implied range. Now our implied range is 0.15 to 0.24, which is way bigger than this implied range. So placeholder, 0 as a placeholder, it's important. Here, target's the 9, alright? We have a 5 after it. There's nothing after that 5, so we leave it alone. 0.2789. Our implied range, again, is going to be what captures that rounded value, 0.27885 to 0.27894. Lastly, we're going to the thousands place. Rounding there. Next digit's a 9. We round up. So 126,000, that's our rounded number. Zeros are placeholders, again, because we're in the thousands. Make sure we have the correct number. Our implied range still goes to that next digit. So this next digit is that 0. So our implied range is 125500. So 125,500 up to 126,400. That's our implied ranges. Hopefully you feel comfortable with this. This will be on quizzes. This will be on exams. It'll be rounding practice. So make sure you know how to round. Make sure you know how to do your implied range. Are we going to round as we calculate means and standard deviations and variances? Yep, we are. So if you don't know it, if you didn't know how to round, now's your time to learn. Can we round in R? Oh yeah, of course. There's several functions that we can use. The primary function that we use is round. All right, and round function takes on two objects. So we're going to give x, which is a value. That's the number that we want to round, and then we have to tell R how many digits we want to use. Now the digits here refer to decimal places. All right, digits here refer to decimal places. So if I have 2.567 and I want to round to this place, then digits is equal to 2. So it's the second decimal place. If we want to round here, let's say, all right, or here. Now what we're going to use is negative numbers. So this is kind of tricky to know. It's good if you round it in your head first and you know what number you should get, and then you just adjust the digits. So what R does is, if you leave it off, the default is 0. 0 digits in your decimal place. That means we're rounding at the whole numbers. So 0 is at whole numbers. If we have 1, we round first decimal place, 2 is second decimal place, 3 is third decimal place. Alright, so we said 0 is at the ones. If we go -1, we go to the left of that, so we're at the tens place. If we go -2, we're at the hundreds place. If we go -3, we're at the thousands place. So you should see that when we round, and we're rounding to different points, we're going to change the digits. That tells us where we round with the digits, and it gets a little bit tricky, but again, if you round it in your head and you know what answer you should get, then you can just adjust the digits until you get the answer that you're looking for. Two other ones that we'll talk about and we won't really use a whole lot is ceiling and floor. Ceiling and floor takes on the same type of variable, so just a single number, and it will round that number. Ceiling rounds it up to the next whole integer. Floor rounds it down to the whole integer. So if we have 1.001 and we use ceiling, that rounds it all the way up to 2. If we have 11.11, it rounds us up all the way to 12. For floor, it rounds us down to the whole integer. So 1.001 gets us to 1. 11.11 gets us back to 11. Alright, so here's your turn to practice in R. What I want you to do is round these numbers to the digits that I specify. Now here I have underlined the digits as well that is our target digits, and I'm going to try to do this on quizzes and exams along with this column to help you know where to round. I don't need to know the code. This is all going to be in your R script, so you can just go ahead and use the code, get the rounded number, and then also do the implied range. At this point, I would start a Markdown document for this presentation and start with rounding and just kind of annotate and say I'm going to round a specific number to this certain value. Here's the code. R will give you the output, and then you can give the implied range. So pause the video, open up R Studio, and round these numbers. Alright, so hopefully you paused the video, you have R up, and you worked on it. So let's do this. So here's my R Studio. We're going to work it through. So I made a new R Markdown document. I do things with PDF since I don't have Word on my computer. If you use a Mac, you can also use Word, or you can do a PDF, but I don't have templates for PDF. I haven't figured those out yet. So I'm just going to point out this part up here. That will automatically generate the date when you create the Markdown document. So I'll probably start including this in all of the Markdown documents that I include. You can do the same, or if you just want to manually specify the date, then you can. It's up to you. Alright, so here's my Markdown document. I've already went ahead and put these in so that you wouldn't see me type, but I started out with the first one. So my first heading is rounding. Next heading is rounding in practice. That's heading number 2. And then what I'm going to do is use my fourth heading, number 1. So we're going to round the numbers to the appropriate digit, state the implied limits. So number 1, 15.679. We're going to round it to the hundredths digit. So I'm going to create my chunk, and I'm going to use round. We're rounding this 15.679 digits. Well, it's 2 decimal places, so digits equals 2. I run it. I get my rounded value, 15.68. Now I can look at it and say, yeah, that's what it should be. R agrees. The implied range 15.675 up to 15.684. So again, it's just basically drop the number down, drop the target number down 1, add a 5 after it, leave the target digit alone, add a 4. That's our implied range. Alright, next one. We're going to round to the tens digit. Alright, so we're going to add a chunked. Now I do Control-Alt-I to insert, but you could have also done this, Insert R, to add the chunk. So we're going to round 179.55 digits. So we're going to the tens. If we leave digits equals 0, it rounds to the ones place. So to get to the tens, it's 1 to the left of it, or -1. If I run it, I get 180. Does that make sense? Yep, it does. So the implied range here, remember the 0 is there for the placeholder, so it's 175 to 184. That's our implied range. Next up, make a chunk. This time I'll use the insert. We're rounding, lowest integer, so we're going to use floor. And you don't have to use anything else. Just use floor. It'll automatically take it down to the lowest integer. Now with the implied range, we do the same thing. 1.5 up to 49,332.4. You'll know that this might not capture the actual range, but we didn't round it normally. We took the floor. Alright, highest integer. Highest integer is ceiling. So we're going to take ceiling of 3.679, run it, goes up to 4. So our implied range, 3.5 to 4.4. Fifth one, we're going to go to the 10,000th digit. So round 1.796832 digits equals, that's our fourth digit, fourth decimal place. And we get 1.7968. So our implied range, 79675 to 1.79684. 1.79684. Alright, and last, to the hundredths digit. So round 38962.2 digits, hundreds digits. So at that 9, that's what we're looking for. 0 goes here, -1, -2. And I will run it. Just remember, Control Enter will run the line. So 39,000. Now that rounded up, and remember, this is tricky, but we are doing implied ranges of 39000. So our target was here. We go to the next digit. So what we would do is 389,500 up to 39040. 3895. I got one too many zeros. 38950 to 39040. All right. So there's possibilities that our implied range won't actually capture the number, and it's because of our rounding rules. And that's why the textbooks kind of made the odd versus even distinction. So when the implied range doesn't capture the number, really it doesn't when that next digit is a 5, then what it wants is have equal number of implied ranges that capture it as that don't for that value. For us, we're not going to worry about it. So that's our implied ranges. This is how we use R to figure them out. All right, derived variables are going to have their own discussion here. So derived variables are variables that are computed from two or more independently measured variables whose relations are expressed in a certain way. So we will go out and normally on the smallest sampling units we will take multiple measurements, which means we will have multiple variables. Each of those variables might only capture part of our process of interest. All right, so a single variable only partially explains or accounts for variation of our target. So what we can do is utilize all of those variables that account for partials and create a derived variable that tries to capture the overall target. So the example that we can give is like overall healthy size. All right, that's what we hear about in the news. Well, we can use weight, and weight captures some of that because heavier people tend to be unhealthy. Lighter people tend to be more healthy, but our problem is that weight and height are correlated. So taller people tend to weigh more, shorter people tend to weigh less. Just because you weigh more doesn't mean you're unhealthy, especially if you're 6'5". So what people have done is come up with this index, the body mass index, to try to capture the overall healthy size. So they use both weight and height to calculate it. Now we can say, does BMA actually capture overall health? I'd argue no, not quite. But it's an index, and it definitely accounts for more variation, or it's a better descriptor of healthy size than just weight alone. Another one is perhaps we're interested in light capture of leaves. How well leaves can capture light. We can look at length of the leaf. We can use width of the leaf. Both of those kind of imply that larger leaves. But you can have a long leaf that's really thin. You can have a fat leaf that's really, really short. All right, and the length and width don't necessarily capture or tell us the entire story. So what we typically do is utilize surface area. And surface area uses both lengths and width. So surface area here would be considered a derived variable because we measured length and width directly. Now that could change because, again, you can take an imaging program and kind of outline the leaf itself, and the computer automatically calculates the surface area, but surface area still includes length and width. Where does this fit in for statistical analysis? Well, if you are comparing the light capturing ability of two different species of leaves, you would use surface area to do that. And when you use surface area, you don't include length and width in that analysis. Surface area already includes these variables. So what types of derived variables do we have? Well, we've got three types. We've got ratios, indexes, or indices, and rates. A ratio is just that. You take two different variables and you make a ratio and report it as a single value. Sex ratios, surface area to volume ratios, variance to mean ratios. These are all ratios. They typically have a ratio in their name. When working with ratios, we typically scale it to 1 and then express it as a single number. So with 59 females and 48 males, we don't say that the sex ratio is 59 to 48. What we normally do is do 59 divided by 48 to get a number. So if you did 59 divided by 48 on your calculator, you would get a ratio of 1.23. We would say sex ratio is 1.23 females to every one male. Makes things a little bit easier to work with. We can also usually express ratios as a percentage. Percentages are a type of ratio, especially when dealing with sex ratios. But not all ratios can be expressed as percentages. So when we talk about percentage female, percentage infected or uninfected, those are ratios as well. Second derived variable is index. These are often generically used to refer to derived variables. So a surface area to volume ratio could also be called an index, and some people would do that. I prefer to kind of restrict the index, the usage of the term index, to either standardized variables, summing a sum of some sort of scoring, or perhaps when we use a function, some sort of equation that uses multiple variables to come up with a single derived variable. So I think index is I think better describes that derived variable. So what's a standardized variable? It's a variable that we tend to standardize to another one. So if you're looking at weight, what we can do is take weight and divide it by the height of the organism, and that's like a standardized weight. What we've done is control for the differences in height. Combining the variables, let's say you figured out a best fit equation. You use that best fit equation to make a prediction, and then you analyze that predicted value. What we've done is combine variables into a single variable itself. That could be an index. And but normally how you would work with ordinal variables, especially with a multi-question questionnaire, is that you can take the score on certain questions and add them together to get like an overall satisfaction score. All right, so if you add up all of those scores, that ends up being an index. Rates are just that. It's going to be some sort of count or measurement, and it's going to be per unit time, area, or volume. And the per time, per area, per volume is always going to be natural. It won't be some arbitrary time or space interval. So speed, that's natural. It's a rate. Miles per hour, miles per kilometers per hour, meters per second. That's all natural. Yeah, we arbitrarily defined the time unit, but the speed was still a natural unit. It was per something. Total time to complete the Indy 500, though, is actually a ratio, because we've arbitrarily defined the distance, and then it's basically time for that distance, so it's no longer a rate. Do we need to really worry about ratios, indices, and rates a whole lot? No, not really, because they're all analyzed in the same way. So when it comes to analysis and it comes to providing descriptive statistics, you don't necessarily need to figure out, am I working with a rate or am I working with a ratio? Now, all of the assumptions of our tests have to be true, but in a lot of cases, at least in this class, a lot of our assumptions will be met. We will talk about what to do when we violate assumptions. So, if there's really no difference, why would we use no difference in analysis? Why would we use derived variables? Well, the advantage of using derived variables is when our individual variables don't explain enough of the variation of in our biological process of interest. So, light capturing ability. Length and width, they capture some of the information, but they don't capture enough. So, doing a surface area or if we were interested in thermodynamics, so heat loss, surface area to volume ratios captures more than just surface area or volume alone. So in that sense, yeah, ratios make sense. We're gonna have more explanatory power if we combine the individual variables. Disadvantage, though, is that these measures tend to be relatively inaccurate and imprecise. And it's because our implied ranges start to carry over with each other. So, as an example, I've got surface area to volume ratio, and I have a surface area. So surface area to volume is a ratio, all right? And let's say we have 3 over 5. That's our ratio. Surface area 3, volume is 5. That gives us a ratio of 0.6. Now, if we report this, it looks like our implied limits is 0.55 to 0.64, and you'd say, yeah, that's kind of how we would round our numbers. But that's not reflective of the error, because this error is only reflective of that final number. We have to account for the error of both the 3 and the 5. So when we do that, we have to look at the implied ranges of both of these values. So the implied range of 3 is 2.5 to 3.4. Implied range of 5 is 4.5 to 5.4. All right. If we want to know what's the smallest number we can get, then what we can do is take 2.5, it's the smallest up at the top, and the largest at the bottom. That gives us the bottom measure of our error. The largest measure of our error is 3.4 divided by 4.5. That gives us the largest value, and what we can see is that our surface area to volume ratio now has an error of 0.46 to 0.76. That's much, much larger than the implied limits that we would have come up with for this rounded value. So we go from basically 9 units for each of our measures up to 30 units difference. Second example is the index. So we did first ratios, now we're going to do multiplication. So if we did surface, if we had surface area, that's going to be length times width. So if we had 3 times 5, well, that gives us 15, and this would be the implied range if we assumed that 15 was a rounded number. But that doesn't really reflect the error. The error accounts for both of our implied ranges. So again, 3 has the same implied range as above, 4 has the same implied range of above, and what we're interested in is what's the smallest number we could get. So 3 could be 2.5, 5 could be 4.5. If we multiply those two smallest values together, we get 11.25. If we do the largest, we get 18.36. So you can see that our implied range went from basically 1 unit, half a unit below 15, or 0.5 below 15, 0.5 above 15. All right, but now our actual error is much, much larger. Now from 15, we're talking about almost 4 units below versus 0.5, and 3 units above, which again is much larger than 0.5. So just something to kind of realize that when you work with derived variables, you can have increase in error rates, and it's something that you may have to consider. So these variables relatively inaccurate. We have much higher error rates. Number 2 is that they typically do not follow a normal distribution, and if they don't follow normal distributions, then we normally can't work with normally distributed data. Now, is that going to be a huge problem? No, not really. We can kind of figure that out ourselves. Number 3 is that these derived variables, while they can capture the biological process of interest, they don't really tell us anything about the relationships between the two individual variables. So surface area to volume ratio, while it gives us the ratio and it tells us tells us some information about heat gain and loss and water loss and so forth, it doesn't really tell us anything about the actual surface area or the volume itself, and it doesn't really even explain the relationship. So we don't know if surface area increases as volume increases, or if maybe surface area increases faster than the volume increases. Oftentimes it's better just to work with the individual variables themselves. And lastly, frequency data or counts might not generate a continuous distribution. And this comes down to, can you get all of the data points? So, for example, some numbers are going to be impossible to obtain. So let's do a dice example. You roll a single dice, and that dice can have any number from 1 to 6. If you take 2 dice and try to do a derived variable by rolling them twice, rolling 2 of them together, the numbers that we can get is 2 to 12. So we have lost the 1. We can't roll a 1 anymore. So that number is completely impossible to obtain. Also, when dealing with if we look at the probabilities, some of our values are going to produce the exact same ratio, or in our dice example, the exact same value. So there's only one way to roll a 2. There's only one way to roll a 12, but there's 6 ways to roll a 7. And that can be problematic, because again, it doesn't really give us the whole story about the individual variables themselves. Now, if we do have frequency data, one thing that we could do is transform the data to using probits, which is an inverse normal probability, to kind of fix some of the problem. Are we going to do this? No, but you might need it for a graduate project or for an honors project. And if you do, then I can help you out, and we can kind of go a little bit more detail of what is happening. All right, so now what do? We collected data, we have it in Excel, we've imported it into R. Next question is, what do we do now? Well, one of the first things that I like to do is to make a display, make a table, or make some sort of graph. And this is for a couple reasons. So first reason is we want to look at the underlying distribution. Does it look normally distributed, or did the data look skewed? Does it look like the data have a single peak in it, or are there multiple peaks in the data? And most importantly, making these graphs or tables tell me if I made a mistake of data entry. So if we're reporting means, let's say if we're reporting lengths of, let's say, snail shells, our values could range from, let's say, 5.5 millimeters up to 13 millimeters. But one of those, you forgot a decimal place. So one of those has, let's say, 100 millimeters. Well, in a data set of 1,000 data points, it's hard to find that 1,000 data point, that 1,000 value, unless you make a histogram. And then all of a sudden you'll see you've got this one point way out far in the hundreds, and you're like, I made a mistake, let me go find it, and then you can correct it. So there's utility to making a graph or a figure as the very first process. What we're going to do is learn how to do histograms and tables and bar graphs now, so that as we move forward, you've already learned how to do them, and you can make them, hopefully have an easy time at making them. Alright, so frequency distributions. These are arrangements of the classes of observations with the frequencies of each class indicated. Now our frequencies can either be a raw count or they can be a percentage of the total. We can illustrate them as a table, which would take on the name as an enumeration table or a frequency table, or we can display them as a figure, so either as a bar graph or a histogram. What we're going to do is start with our frequency tables. So we've measured data, it's qualitative in nature, so it's going to be categorical, and we get it into R. First thing we want to do is look at it. Do our numbers look correct? So our example data set is posted. It's qual freq table data, so qualitative frequency table data.csv. What I want you to do is go ahead, download it off of Blackboard, import it into R, and if you want everything to match, I want you to call it qual data. So pause the video, download the data set, and import it into R. Alright, so hopefully you've imported the data set. I'm going to do this here. So we are moving to frequency tables. So generating frequency displays. This is qualitative data. That's our first idea. And what I'm going to do is reading in the data. Alright, so I've already downloaded my data set. So input text from base. I'm going to go to my folder, R data, qual, frequency table data. Open it. I want it to match, so I'm going to do qual data. I see my input file. I see my data frame. Make is in bold. It looks correct. So I will hit import. Imports it. Very important to do. I'm going to take that, copy it, and paste it. So now, if I go to knit this, R is automatically going to read in that data set, so qual data is now accessible. Alright, so we've read in our data. Now, I want to make a frequency table. Alright, the frequency table is pretty easy. Alright, so the function that we're going to use is table and x, which is a vector. That will automatically go through, figure out what categories are in our data set, and start to count up how many items actually exist with that category. So for this qual data, we are going to do table. Now, if you do table and qual data, that's a data frame. That's not a vector. And what will happen is that, yeah, you will get a table. But if you have another variable in that data set, you will get a really long output. So what we need is that vector. We want that variable. So as review, we want to do a table of make. So we have to tell R where to find make. Where is it? It's in the qual data. So we will use the $sign to specify or to tell R that I want it to make a frequency table of make where make is found in qual data. And when I run it, it makes the table. Now note the change, it took out make, and that's because that's how R will make tables of data frames. And we can try it with another data set so you can see what I'm talking about. But frequency table, qual data $sign make. Now we get this. That's our output. Is that nice? So it's the same output. Is that nice looking data? No. It's just the frequency count. It's just that. So how do we go about making a very nice table? Well, now we have to import it or get it transferred over to Excel. So what I would do, here is LibreOffice. It's similar to Excel. I'll say it's a free version. All right. So if I were to make a table, what I'm going to do is do it in Excel. All right. So my headers, make and count. I've got Chevy, Ford, GMC, Toyota, and other. All right. So I've changed the order. Other appears first. Before Toyota, I want that other to appear last. And then I'm just going to enter in my numbers. So I've got my table. Get everything centered. I like to bold my headers. It's optional. And then it's important to do the lines. So I've got lines. I've got up here, if it's bigger, I've got borders all set up. But in Excel, what you can do is right click and do format cells, and that will open up this just like Excel, and there's a borders tab, and then from the borders tab we can add a top and a bottom to the header row, and then a bottom format cells to the table. Alright, so couple rules. Let me move this up. Of table creation. We want a logical order. So I did alphabetical going down with other at the bottom. Does it have to be alphabetical? No. It should just be a logical order, and that's up to you how to do that. Number 2, you want a line at the top of the table. So there's our line at the top. We want a line at the bottom of the table. So it kind of sets off the table and says, here's the start, here's the bottom. When you have a header row, you also want a line to separate the header. What we don't have are lines in the body of the table. So notice the lines here. We don't do that. The only time I'd put lines in the body is if we have, if I did something different and said, let's say US made or imports. And then I might put a line that separates US and imports. You'll see it in the journals. Just kind of depends on the table. And then finally, if you have a total row, then it should go at the bottom. Now a total row is optional. I'm going to put it here, and I can add it up by hand, or I can use the sum function, so equal sign sum, parentheses, and then highlight, and when I press enter, it automatically adds it up. Now if you add the total row, make sure it's centered, and make sure that we have a line both at the bottom of the table and between the total row and the body of the actual table itself. Am I going to test you on making tables? No. I'm not going to test you on making tables in Excel. I'll test you on making these frequency tables. Why does it matter? It's because when we get ready to do fitting distributions or doing chi-square tests, we can use the table function to generate this table, which then gets fed into the chi-square function to do our own tests, so we don't have to manually do it by hand. All right, so that's qual data. One thing that I did use was the head function. I did that when I initially made it because I wasn't really using RStudio, but I used it so that I could see what my variable names are. I did that just by doing the drop down arrow. So I did that and I saw that it was make, so I used that. But if I didn't, what I could do is this. I can do head dataset and run it. And what that'll give you is the top of the data frame. It could be the top of a vector if you have a vector too. Basically gives you the first 6 rows. And I often use it to check whether or not it imported. Now, with the R markdowns, I normally don't. I just drop them down. But if I go over to any of my other scripts, then yeah, I often use head all the time. So I can add it in here just to make a note that says head data. This gives the first 6 rows of the data frame, just so you can, it's useful. It's a useful function. Alright, so that's our frequency table. Can we use the frequency table with quantitative data? Yeah, depends on what type of quantitative data we're working with. So here's a quantitative data set. It's quant freq table data. So pause the video, download the data set, read it into R as quant data. Alright, so hopefully you've imported it. I've already imported it. So what we are going to do is I will go up here because I'm going to import the data set and I want it to appear in the same place. So I've got R data quant frequency table data. We're going to do quant data import. There's my code. Copy it. Go back up to here. Quant data. And I am going to use the head function again, just to confirm. So I will run the chunk. There's qual data. Here's quant data. Alright, so quantitative data, this data set represents a data set. And some of you may have had this lab, but we would go out, have a grid, and then drop the grid out in our area and count how many trees or how many plants are inside that grid. And then we get a new place, drop the grid, count how many plants are in that grid. And what we end up having is a bunch of grids with numbers that say how many plants are in each grid. So that's what this data set represents. So if I look at the quant data, I've got 800 rows here of data. And it's zeros, ones, twos, threes, fours. So basically it says all of these zeros, these are all grids that had 0 plants in it. And then all of these grids have 1 plant in it. Alright, so it's a vector. But what we want to do is try to summarize it. So what we're going to do is use quantitative data. Quantitative data. Alright. I guess I should have put reading in the data here. Reading in data. There's my data set. Alright, so let me take that out. Alright, so now what we want to do is do a frequency table. And frequency table is done the exact same way using the table. We need to give it the vector. So our vector found from the head function is plants. I can do the drop down, it's plants. So I am going to do table quant data $plants and run it. And I get my frequency table. So what this tells me is that I have 412 grids that had 0 plants in it, 163 grids that had 1 plant in it, 104 grids that had 2 plants, 46 that had 3, and so forth and so forth and so forth. Alright, I can see I didn't have a mistake. I don't have, 100 and then 1, which would be like one grid has 100 plants in that would have been a mess up. Alright, so it looks like I have the correct data in. Now with these frequency tables for the quantitative data, the same rules apply. I take the data, I put them into Excel, and I set it up the exact same way. So it's a logical order. In this case I use 9 or more because there is a possibility that we have more than 9 plants in a grid. I give the frequency. I have lines at the top, I have lines at the bottom. I've got a line that separates the header from the body. I don't have a total row because it doesn't really make a whole lot of sense in this case. I'm reporting on the number of grids that have a certain number of plants per quadrat. So I can give it and say I can give it in the text of the manuscript or in the table legend and say we had 800 grids. That's the sum. So total doesn't really give us any more information. But with these discrete data, we can do a frequency table and we make it the exact same way as if we had qualitative data. If we didn't want to do a table, if we wanted an actual graph, one thing that we can make is a bar graph. So a bar graph should only be used with qualitative data. Let me repeat that. We should only use a bar graph with qualitative data. Why? It's because the bar graph, the height of the bars, the width of the bars, or actually the area of the bars, is what conveys information. And it conveys information on frequencies. When we work with frequencies, we always start at 0. If we use bars to represent means, most cases, those means we're not starting at 0. So we're relaying false informations. So these bar graphs really represent counts and frequencies. How do we do it? Well, we do it with the bar plot function. Alright, so we're going to go back. We made a frequency table of our makes, and I'm going to add this here. I'm going to make a bar graph. The function of the bar graph is bar graph and then table x, where x is a vector. Alright, so what bar graph is going to do is to take the table that we give it and make a bar graph. Why? Well, let's check the help file. I'm going to go over here and type in bar graph, or bar plot. Ooh, yeah, bar plot. That's the function, sorry. Bar plot. The bar plot takes height. So what R needs to know is the height of each bar. Well, what is the height of each bar? It's the count. It's the frequency. So in order to make a bar graph, what we're going to have to do is give it the frequency of each of the categories that we have. That was obtained from our table. So the function is bar plot table x. Control-Alt-I, bar plot, table, qual data, $make. And I'm going to make it. There's my bar plot. Now, here's the thing. Does this bar plot match what is in the presentation? So that's in the presentation. That's what I made. Let's get this shrunk down so you can see it. Oops. A little bit over here. Side by sides. Pretty close. Is that the same? No, it's not. The bar graph should follow the same rules as the table. We should have some logical order, and in this case, our logical order is the makes, with the other category being less. R didn't do that. R put other before Toyota. Why? It's because R alphabetizes everything. So what we have to do is tell R how we want our variables to appear. All right, so what we're going to do is factor it. All right, we're going to factor our variable. Well, what is that? Well, let's look at the help file. Factor is used to encode a vector as a factor. Terms category and enumerated type are also used for factors. All right, if argument ordered is true, the factor levels are assumed to be ordered. For us, we're not going to really specify order. All right, we don't have to do that. I think that's the default. But what we are going to do is specify the levels. All right, so what the factor function is going to take is a vector. So our vector is qual data $make. And what we are going to do is specify the levels in that factor. So the levels of the factor are Chevy, Ford, GMC, other, and Toyota. How do I know that? Well, I can just look at it. So if I do levels, qual data make, and run it, I get Chevy, Ford, GMC, other, Toyota. Alright, so those are the levels. I want to specify the actual order. So I've got Chevy, Ford, GMC. I want Toyota, and then other to be last. Alright, so that's going to factor it. Now if I run it, whoops, what happened? There it is. Alright, it factors everything. So I didn't run the bar plot, I just ran that one line. It factors everything and basically says, okay, yeah, the Chevy's the Chevy, the Ford's a Ford, GMC's GMC, everything. But note the levels. So the levels that gets reported is Chevy, Ford, GMC, Toyota, other. If I check levels, again, I'm going to put it here, it didn't correct it. So even though I had it correct up here, it still didn't report it here. Why is that? Well, I didn't save it. So what I want to do is factor the make vector, the make variable, with this order, and then save it back to itself. So I do that. It factors it, saves it to itself, and now when I check the levels, now it's fixed. So when I make my bar plot, I've replaced the order. So this is going to be very useful, this function. This is going to be very, very useful to know. So function factor vector and levels equals C quotation marks comma quotation marks comma quotation marks and so forth. We'll do A, C, and B just as an example. So what this factor is going to do is refactor a categorical vector, categorical variable, and specify that the levels are ordered A, C, then B. Alright, we'll use this again. We'll use this with making figures. So if we do treatments, so we've got a control and then treatment 1, treatment 2, treatment 3. We normally want the control to be first or last. All right, but if the terminology, if the names put the control in the middle, we're going to use factor to make sure that the control gets listed first or last. All right, so that's a bar plot. Also possible to make. What about quantitative data? Well, quantitative data doesn't really make sense to do a bar plot, and here's why. All right, so I'm going to do histograms here and just kind of introduce the bar plots. So if I did bar plot, quant data, $, plants, and run it, I get something like this. Oops, sorry, table. Forgot that. That's not right. There it is. Now we get that. We get something like this. Now this looks... Oh, sorry. That's it. I'm working on my other screen. So if I make a bar plot of these quantitative data, it's not going to be representative of the underlying data. So I'm going to label this histogram because that's what we're going to do. So we want to do a bar plot, table, quant data, $ plants. When we make this bar plot, this is what it looks like. This is similar to the histogram. Let me move this over. This is similar to the histogram. You've got the same shape. I mean, it looks like it goes down. That's fine. But notice the break between the bars. All right, that break implies that we have breaks in our data. And while we are working with discrete data, and they can only take on certain names, we're still working with numeric data. It's still a scale. So the bar plot kind of implies that each bar is completely independent of all other bars, but they're not. In order to get to 3, we had to go through 1 and 2. So a bar plot for a histogram doesn't really make sense. We really need the bars to touch, and that's going to really emphasize the measurement aspects of our data. So how do we do that? Well, pretty easy. We use the histogram function. All right, so the function that we use is hist and x, breaks, where x is a vector, breaks is number of bars. Not quite the number of bars, but that's how we would adjust the number of bars. So what I like to do first is just do hist. We're doing quant data, $ plants. Run it, and I get my histogram. R does a really good job of making histograms, by the way. I only have to adjust the breaks for certain data sets. So here it is. It relays a similar type of information as a bar graph, but that extra bit of information that the bars are touching really emphasizes that we're working with a measurement type of variable, a count variable, that the numbers just go from one to the next, so that the bars are not independent of each other. So histograms are going to be used for quantitative data. Really explain the frequencies and really show the frequencies. Now, how do we make these frequency distributions? Well, if we have qualitative data or discrete data and we want to make a table, then we just use the table function. It's pretty easy, self-explanatory, hopefully. And normally our data is going to be such that the table function works, no problem. Some data tables are going to be way too big, and then you have to start combining data sets. So you could have a situation where some of your groups have one or two counts, and then other groups are like 50 to 100. Well, then maybe you can look at some of those less represented groups and combine them into one. If we have quantitative data and we want to make a frequency distribution, then my suggestion is to use a stats program like R. Why? It's because in order to make the frequency histograms, we have to take our continuous variables, our continuous data, and put them into groups. That is called binning them, because we're putting them in bins, the groups, the group of data. And those groups, those bins, are going to be the bars in the histogram. The number of bins that we have in our graph determines the width or the class interval. So it's basically the size. Is our bin going to be 1 to 2, or is it going to be 1 to 1.5, and then 1.5 to 2, and so forth? Normally, when we make these histograms, we want 12 to 20 bins, so 12 to 20 bars. And we want our bars to represent reasonable class marks. So we want them to be natural breaks. We want it at 1, 2, 3, 4. We don't want it at like 1.1 and 1.8 and 2.1 and 2.8 and so forth. We want it to be easily to view, easy to conceptualize in our brains. What about this 12 to 20? Well, if we've got too few bars, too few groups, then our histogram's basically going to be flat. We're not going to see any detail. If we have too many, then we're going to see way too much detail. It's going to be messy, and it's going to hinder our ability to really determine if our data have a normal distribution, an underlying normal distribution. So with our quantitative data, we're just going to let R do it for us, and R does a pretty darn good job at it. So here's the function again. I kind of included it in the script so that we can work with it. So here's our example of a breaks. Here's the code used to generate these different figures. I'm going to generate it here in my Markdown document. So Histogram Example. So I'm going to start out with X is our norm. What this function does is generates a variable, 1,000 data points. So just going to annotate this. We're going to generate 1,000 data points pulled from a normal distribution. Alright, so R, for our distributions, R is random. Norm is the distribution, normal distribution, and we want 1,000 data points. And we're going to save it, those 1,000 data points, as X. And then we're going to do our histogram. So that generated our data points. And then we're going to make our histogram. So first up, hist X. That looks pretty good. R does a good job. You can see the peak. This is a normal distribution, so we should see kind of evidence of a bell curve, and that's what this looks like. Now, is this exactly what I got in the presentation? No, it's not. And it's because of the random number generations. When you do this, you'll probably get a different graph too. But we can see that we got this smooth up to the peak and then smooth decline to our tail. What if we weren't happy with this? What if we wanted to have more bars? Well, I can do that by specifying breaks. Now, I can say I want 4 bars and it's going to work because I've worked through it before. So we've got, that's our first one. That's our one that we wanted to see. Here's 4. Notice the detail that was lost. Now we've got 2 bars up at the top, 2 really low bars at the tails. Now we could say, oh yeah, it goes up to a single peak and goes down, but does it really? How do we know that we don't have an increase that goes up, goes down to a valley, goes back up, and then goes down? We don't know that. So having too few bars really hid some of the data, some of the patterns in the data from us. What if we use too many breaks? well, then it just becomes messy. Then it just becomes messy. So there's our first one, there's 4, here's our last. Notice all the bars. You can see even in the middle, right around our means, we've got some low values. Well, that's just random chance. Now, when we group them and start binning them together, yeah, we take on this curve, and you can say, yeah, this kind of looks like a normal curve, but there's a lot of variation here. You might be tempted to say, yeah, this isn't normally distributed, but I think you'd be wrong. There's just too much detail. It's too messy. Just a word. I said I wanted 4 bars. Let's say I wanted 5 bars. There's no guarantee that that's going to work. All right? There's no guarantee. I specified 5 bars, but look, I got 8 bars instead. So just because you tell R how many breaks you want doesn't mean that you'll actually get it. R's still following a process to make some pretty figures, a pretty graph. So if it determines that it can't really do 5 breaks or 6 breaks or 7 breaks, then it will go either higher or lower to make sure that the figure looks okay. Alright, so that's histograms. Alright, so now just for practice, and for practice in R, what we're going to do is make our own frequency distribution. And this is the steps that R is going to follow. So R is going to take the data that we give it. It's going to group the observations into classes. It's going to figure out the classes. It's going to figure out how many individual data points fall within each class. It's going to decide if we're going to use a midpoint, left or right classification. So is our label going to be referred to the midpoint of that class? Is it going to be the left edge of the class? Is it going to be the right edge of the class? And then once it has that, it's going to make the table or figure. So R does this all in the background. If you do it by hand, you have to do all of this stuff, which is tedious. So for our example, I've got these vectors. I want you to enter this into R as a vector and then make a histogram. So go ahead, pause the video, make your vector, make the histogram, and then I'll come back and we'll make it. So you should have been able to get this into R, and it's good review. It's been a while since we've really worked with R extensively. So this is what I did. So first we have to create a vector. So you create the vector with the C, the combine function. So create vector using C function. And I saved it as the vector name vec. Alright. Then once I have that vector, all I have to do is give it the histogram, hist vec. And I get my histogram. Can I change the titles and the labels? Yep, we can. We can do that. If you need to do that, come talk to me. I'll show you how, or you can check the help function. It's main is here, and then xlab for x labels. Alright, so should be pretty easy. What if we didn't have R and you collected those data out in the field? Could we make a histogram and just do a quick comparison? Yeah. Or do a quick comparison if we have 2 groups? Yeah, we can. We're not going to do a histogram per se. What we're going to do is something called a stem and leaf display. This was first described by Tukey in 1977. What it is essentially an ordered array. All right, so it's not necessarily a histogram, but it kind of looks like a histogram. These ordered arrays, the stem and leaf diagram, are actually pretty easy to make by by hand, and R can make them too, so you can check your work. So here's our general method. The method is to first make the stem, which will be usually the 1's digit. So it's an ordered array. First we make the stem, and that's our leading digit, and we put them in order. Then what we're going to do is look at the next value, and these are going to be the leaves. We're going to put the leaves with each of the stem. And then once we're done, we're going to order the leaves. And that gives us our ordered array. So for example, I'm going to show you how to make a stem and leaf diagram using these data. Alright, so we're going to make a stem and leaf display using the data that was in the presentation. So the first thing we have to do is make the stem. And by making the stem, I'm going to write numbers from smallest to largest. So what I can see from our data set is that we have 6.5 is the smallest value, and we go up to 11. So I'm going to start with 6, 7, 8, 9, 10, 11. This is our stem. Those are our stems. And now I'm going to go for the leaves. And the leaf is that ones place, that tenth, the first decimal point. And I'm going to put it with the 7. So the very first number was 8.7. So 7 goes here. 8.5 goes there. 9.4. 10.0. 6.3, 7.8, 11.9, 6.5, 6.6, 10.6, 10.2, 7.2, 8.6, 11.1, 11.6, and 6.5. Alright, so that's our stem and leaf, and now what we do is order the leaves. So I still have the same stems, 9, 10, and 11, and now I'm going to order the leaves in order. So 3, 5, 5, and 6, 2, and 8, 5, 6, and 7, 4, 0, 2, and 6, and 1, 6, 9. This is a stem and leaf display. Notice as you make them, you should try to make sure your leaves all line up in the same place. So graph paper helps to do this, or just kind of try to eyeball it. And this way we can kind of look at it sideways and say, yeah, this kind of looks like a histogram where the bars represent the number of leaves that we have in our display. Alright, so you can see it's really not too hard. And what we can do is check our work by using a stem function in R. Alright, so what I'm going to do is confirm that my hand-drawn stem and leaf diagram matches what R would give. Alright, so first thing we're going to have to do is make our vector. We'll do vec2 for vector 2. Alright, so I am just going to make my vector 8.5, 9.4, 10, 6.3, 7.8, 11.9, 6.5, 6.6, 10.6, 10.2, 7.2, 8.6, 11.1, 11.6, and 6.5. Alright, so I made my vector, and now I'm going to do the stem and leaf display. Now the function is stem and vec2. Alright, and I'm going to run that, and I get this. Alright, so R tries to make things pretty, and by making things pretty it wants to make sure we have an appropriate number of items in each of our stems or in each of our bins. This doesn't look like what we made by hand. What we have to do is adjust it. We have to scale it. So our function is stem x and then scale. Alright? So x is a vector, scale is a number. or an integer. All right, so what does scale do? Well, it's going to either compress our stem and leaf diagram, so start combining groups, or it's going to expand it. So what are some of the values? Well, let's just play around and see it. So if I do scale equals 1, that's what I get. If I do scale equals 2, now what I've done is taken what was standard and increased it by 2. So basically doubled the size. And in this way, I've really spread it out. So now this actually looks like what I made, or what we made by hand. What if I did scale equals 4? Well, now you start seeing that we've added extra groups. And what you could see is that we've started making our stems at like the half point mark. So this is 6 to 6.4. This is 6.5 to 6.9. This is 7.0 to 7.4, and so forth. So we've expanded it, and now we have sets that do not include any of our leaves. So this is what we should do. So that's going larger. What if we go smaller? So 0.5 is half of what it would have made. And what it's done is it's compressed it. Now it's basically done 0 and 1. Now where did 0 and 1 come from? Here. It basically looked at the tens. You've got your zeros, which are everything less than 10, and then you have the other ones that are greater than 10. So for the scale, we'll do, if it's greater than 1, then we expand the diagram. If it's less than 1, so decimal places, then we compress the diagram. Alright, so we had to expand it to make it look like what we actually made by hand. So would you do a stem and leaf diagram just of a single variable? Yeah, you can. You can if you're really curious, if it's normally distributed and you can't wait to get back to the lab or to your office to use a computer. But what it's really good is if you had two groups, and now you're interested in not just the distribution of each group, but also kind of doing comparisons of central tendency or even variation. So you're out in the field, you collected group A and group B for the day, and you're back relaxing, sipping a beer by a fireside because it's super cold outside, and you start contemplating, well, is group A and group B different? And you say, well, I really want to run a t-test, but I don't remember that equation, and I don't have a t-table handy. So what can I do? Well, you can make a stem and leaf display. And what we do is something called a back-to-back display. So now what we have is we have our stem. I'm going to bring R back over here. We're going to have our stem, and that stem is going to be shared by both groups. So we'll have group A or B, one of the groups going to the right, our leaves on the right-hand side, and then the other group, the leaves will be on the left-hand side. All right, so go ahead and do a stem and leaf diagram for group A. All right, and we're going to do a stem and leaf diagram for group B, but this time what I want you to do is to have it have leaves on one side for one group and the other side of that stem for another group. All right, for this one, we now need to make a back-to-back display. So looking at our groups, the smallest number we have is a 24. So 24, 25, 26, and 27. That's our stem. And then I'm going to go through and add the leaves for each of our groups. So we've got 24.1, 24.9, 26.3, 24.5, 24.2, 25.3, 25.0, 25.0, 25.2, 25.6, 25.4, 24.8, 25.1, 26.0, and 25.0. And then for B, I do the same thing. So 25.7, 26.0, 25.6, 25.6, 26.1, 24.6, 26.9, 25.3, 26.2, 25.8, 26.3, 26.5, 26.7, 27.4, 25.1. Alright, so right now I can see that it looks like we are shifted, but we're still not done. I have to make an ordered display. So I am going to order our numbers, or the leaves. And we're going to order it the same way, but this time for the A's I'm going to go to the left. So we've got 1, 2, 5, 8, and 9. We got 0, 0, 0, 1, 2, 3, 4, and 6, 0, and 3. And for B, we've got 6, we've got 1, 3, 6, 6, 7, 8, 0, 1, 2, 3, 5, 7, 9, and 4. This is our stem and leaf display. So here, if we could kind of think about a histogram, it would look something like this, where you can see that let's change the ink, where our means and our peaks end up being or occurring in what presumed to be different places. So it looks like our averages for group A and group B are slightly different. Alright, so that wraps up this presentation. We kind of went over variable types, scales, we did some rounding stuff and implied limits, and then we introduced frequency tables, histograms, bar graphs, and so forth. So this is kind of the basic material. A lot of definitions. You should definitely review the definitions, know the definitions, know statistics versus parameters, know why we do samples instead of measuring a population. You should know how to make a histogram, know how to make a bar graph, a stem and leaf display for the quizzes and exams and so forth. So a question I may have would be, well, do I have to do any of this by hand? Yes. Stem and leaf display you will make by hand, and when you make it by hand, you will probably make it on the exam. So I'll have you make a stem and leaf display on the exam so that you can demonstrate you know how to do it. But I won't ask you to make a bar graph, and I don't think I'll make you do a frequency table on at least a closed book of the exam. For the application part, part of the class is using R and learning R, so yeah, you'll have to make a histogram, you'll have to do a frequency table, and you'll probably have to do a stem and leaf display. So next up is to move to descriptive statistics. So now that we understand, have some of the terminology, now we're going to look at describing the data that we collect. And descriptions being central tendency and location, or central tendency and dispersion.